All right, everybody. Chapter 15 is entirely about the special senses. And so, what are these special senses? Well, in order to think about this, uh, this is um, this is a helpful thing to think about here. Uh, this is the in studying the special senses, we're able to distinguish physical stimuli. Okay, these sound waves produced by clapping your hands and the receptors that are specialized to detect those stimuli. Okay, and this chapter is going to focus on the role these special senses have in detecting stimuli like sounds, smells, light, and then transducing or converting them into action potentials and then interpreting them. Uh, First, we're going to take a look at how the special senses are similar to, and in some cases different from, the general senses. So these are all the chapter 15 sections. We'll talk about an overview of these special senses and their differences between the general senses. Then we'll get into the the special senses themselves. Olfaction, which is smell; gustation, which is touch; the eyes, which are responsible for vision; the ears, which are responsible for both hearing and vestibular sensation, that sense of balance. And then we're going to look at how all of them work together. So the overview of the special senses. What makes them so special? Well, they're named because they convey specific stimuli from specialized sensory organs that are in very specific locations in your head. You can probably list four of them very easily: smell, taste, vision, hearing. But the fifth one is that vestibular sensation, or the detection of head movement and position that helps maintain the equilibrium. So you've already been introduced to sensation, okay? Those general senses in the chapters that we've talked about before in the central and peripheral nervous systems. So before we dig into special senses, we're going to look at how they differ from general senses. So starting at the stimulus and detection of the stimulus. The general senses will involve the detection of a stimulus like touch, pain, or temperature, and as we've discussed, each special sense organ detects very specific stimuli: light, sound waves, head movements, and the chemicals that produce tastes and smells. Now, the structure is also a little bit different. In general sensation, stimuli are detected at the receptive endings of sensory neurons, and these receptors have relatively simple structures, and they're located all throughout the body in both the skin and the viscera. Now, in contrast, the receptors of these special sense organs are not neurons, but instead are specialized cells that detect special sensory stimuli. Okay, and these sense organs then convert these stimuli into electrical signals that are passed on through neurons via neurotransmitters. And olfaction is actually going to be the only special sense in which the receptor cells are neurons. Okay. And then the location is also different. General sensory stimuli are transmitted to the CNS by the axons of neurons located in both the spinal and cranial nerves. But special sense organs are all housed in the head. So only the axons of cranial nerve neurons convey this special sensory stimulus. And so just like I said, you see general senses here on top and special senses on the bottom. Now, special senses have that unique receptor cell that will transduce that signal from a chemical or a photo signal to an electrical signal. And this transduction is essentially, yes, when a physical or chemical stimulus is converted into an action potential that can then be interpreted by the brain. Now, specialized general senses have specialized receptive endings that detect touch, temperature, and pain, And the presence of these stimuli will alter the resting membrane potential of the sensory neurons to produce an action potential that reaches the CNS. Now that is different from these specialized cells. These specialized cells have to convert that signal from, let's say, chemicals that produce taste, light waves that end up producing light, sound waves producing sound, or the movement of your head producing a disturbance in fluid. All of those change the way these receptor cells receive signals, which then enables them to send neurotransmitters to the sensory neuron that will then transmit that signal to the CNS. Except for the case of smell, which has direct sensory receptors on its sensory neuron. These are receptive endings of those olfactory neurons up in your nasal cavity. Now, our olfactory system is what allows us to detect chemicals in the air known as odorants, and these are just chemicals that produce the sensation of odor in our nasal cavity. Now, once they're detected, olfactory neurons will transduce that chemical signal of the odorant into electrical signals that our brain can interpret. And of the 40,000 odorants, about 400,000 odorants, of about the 400,000 odorants that we can detect, about 80% of those are unpleasant to smell. And that sort of speaks to the protective role that olfaction has served evolutionarily, warning us about dangerous smells, especially smoke. And in addition to identifying odors, the nervous system also allows us to experience a very specific emotional response that may accompany these smells. That's why you may have smelled something that you've not smelled in years, and it can trigger a memory. One of those is wood shavings for me. I don't know about you guys, but I know everybody has one of those smells that will trigger a memory, a very powerful memory, and that's because olfaction and memory are so closely tied. Now, the actual process of olfaction is going to begin in the olfactory epithelium, and this is a small patch of specialized cells located in the superior region of each nasal cavity. This olfactory epithelium is typically yellowish brown and about the size of your thumbnail. And it has 3 different types of cells that are very specific in it. First, you have olfactory neurons, and these are modified bipolar neurons that work to detect odorants. Since they're detecting chemicals, these are those chemoreceptors that we've previously mentioned, and we have about 10 million of these neurons, and that sounds like a lot, but dogs and other animals with keen senses of smells are found to have several billion of these neurons in each nasal cavity, and I'm sorry, in their nasal cavities as a whole. Olfactory neurons will typically consist of an apical modified dendrite, a cell body, and then a basal axon. And so this is a bipolar neuron, and each of these neurons' modified dendrites project into the roof of the nasal cavity. So they're on the superior region of your nasal cavity, and then they go through the roof of it. And at the end, each dendrite will have that expanded tip containing non-motile, non-movable olfactory cilia that increase their surface area available for odorant detection. These cilia will extend into a layer of mucus produced by olfactory glands, and the axons of these neurons are bundled in groups of 10 to 100, traveling through tiny holes in the cribriform plate of the ethmoid bone to reach the CNS. Collectively, these axons form the olfactory nerve, cranial nerve number 1. And they terminate by synapsing on other neurons called mitral cells in the olfactory bulb. And this olfactory bulb is a structure located in the brain just superior to the ethmoid bone and inferior to the frontal lobe of the brain. These axons will leave the olfactory bulb in the form of the olfactory tract, which then transmits the stimuli to other parts of the brain. And that's all olfactory neurons. Basal cells, these are stem cells that develop into olfactory neurons. The truth is that an olfactory neuron has a lifespan of about 1 to 2 months, 30 to 60 days, due to their highly exposed location. These basal cells ensure that continual replacement of olfactory neurons. Think of shark's teeth. Sharks are constantly losing and regrowing teeth. Well, that's what's happening with these basal cells. They're constantly regrowing and growing new olfactory neurons to replace the exposed, deadening ones. You'll then have supporting cells, and these are columnar cells that support and surround the olfactory neurons. They are going to contain that yellowish, brownish pigment that gives the olfactory epithelium its color. So first things first, we start with this olfactory tract leading down into the olfactory epithelium, right? This olfactory epithelium here, this discolored tissue, is where chemicals are received. And you see a zoomed in version over here, and you see the dendrites hanging down, increasing that surface area through this tissue, allowing them to more easily gather up odorants. And they'll convert that chemical signal into an electrical signal, and then it will travel up through this bipolar neuron into the olfactory bulb right here above the cribriform plate, and then that will transduce the signal down to other parts of the brain to be integrated. And here you have an actual scanning electron microscope view of the actual epithelium. You'll see these sort of, I want to say starfish-like structures, and those are the dendrites. They're all reaching out, increasing that surface area, awaiting chemicals to latch on and be detected. Now, odorants are typically carried into the nasal cavity through the air, but very few of them are going to reach the olfactory epithelium during normal breathing. But what is your normal response to smelling a faint odor of something? You're gonna sniff or inhale more deeply, and this draws in more air at a faster rate and forces it across your olfactory epithelium. And then when odorants come in contact with this epithelium, they're detected by those neurons, which then begins that series of action potentials. So how does this work? How does this take place? Well, first, you have to activate the olfactory receptors. And this happens when odorants reach the olfactory epithelium, binding to odorant binding proteins, which are going to sort of transport them to the receptors on the olfactory cilia. When they eventually do reach the receptors, when the odorants finally reach their receptors, what's going to happen is a chemical reaction, a very specific protein activation pathway. The binding of an odorant to its receptor activates what's known as a G protein. Okay? This activated G protein then detaches from its receptor. This activated G protein then triggers the enzyme adenylate cyclase to convert ATP into cyclic AMP, okay, or CAMP. And when CAMP opens ion channels, that allows sodium and calcium ions to enter these cells. And so the entry of these ions creates a local potential that depolarizes the membrane of the olfactory neuron. And remember, you have to get enough local potential to overcome that threshold. And so that will summate, and then the threshold will be reached at the axon hillock, and boom, an action potential is triggered. So here you see what I'm talking about. This odorant here, this chemical here, is going to bind to a protein in the receptor site. And then what's going to happen there is this receptor is going to activate this G protein. This G protein is going to detach and activate adenylate cyclase, okay? And adenylate cyclase is now going to convert this ATP, this adenosine triphosphate, into cyclic AMP, adenylene monophosphate. And so this CAMP is going to then open ion channels that allow calcium and sodium. They try to flood the cell with positive ions to get it above negative 55 millivolts. And if they do that, enough local potentials will summate, they'll combine, remember, and that action potential will reach the axon hillock, and then it will travel down the length of the axon, becoming an action potential proper, traveling throughout the rest of the neurons. Now I'm going to point out that not all chemical odorants actually bind to olfactory neurons. Ammonia and chlorine, like really strong bleach, instead bind to branches of the trigeminal nerve in this nasal lining, and they produce that burning sensation that you may have gotten if you've ever gotten a particularly large whiff of like a window cleaner with lots of ammonia in it, or if you're doing laundry and you're using some pretty strong bleach. These actually bind to trigeminal nerves in the nasal cavity epithelium instead of the olfactory nerves, olfactory neurons. Going back to the olfactory pathway. So we've activated our olfactory neurons. Now what we need to do is detect and identify an odor, right? So first we have to transmit the olfactory stimulus to the central nervous system. And so what happens here is the axons of these olfactory neurons that make up the olfactory nerve carry olfactory stimuli to the olfactory bulb, okay? So remember, these go up through that cribriform plate and they synapse onto the olfactory bulb. The axon terminals synapsing with the dendrites of mitral cells here will trigger local depolarizations, and then many olfactory neurons with the same receptor type will typically end on the same mitral cell. And the same thing happens here. If enough mitral cells have a summation of these local depolarizations, they will trigger that action potential, which will then send its signal from the olfactory bulb down the olfactory tract into the brain. And as I mentioned last time, this is the only sense that actually bypasses the thalamus and synapses directly onto its target, and that is going to be the primary olfactory cortex. Okay, and this is in the temporal lobe, and this primary olfactory cortex is really responsible for your awareness and your identification of an odor. So interestingly, this is also a common location for seizures, which may often be preceded by the sensation of an unpleasant odor. Now, the neurons from the olfactory cortex then go out and synapse onto other neurons in the amygdala, the hippocampus, the hypothalamus, and various other parts of the limbic system. And this is why you have such strong emotional responses to odors, as well as visceral reactions like salivation. You may experience sexual arousal or nausea because these connections also exist between the primary olfactory cortex as well as the cortex in the base of the frontal lobe. And so this is often thought to play a part in integrating olfaction with taste. So again, remember I said strong smells can evoke strong memories, and these are or have strong emotional responses, and this is where that comes in. They contact all of these emotional responsive areas or memory forming areas, and they all work together and they have such a tight connection. It's hard to separate the two. Disruptions in any part of this pathway of transmitting the signal to the central nervous system results in a condition known as anosmia. And this is essentially the lack of the sense of smell. And this may not sound very serious, but it actually can be. People with anosmia may not be able to detect smoke or the odor of spoiled food. It's also possible for these people to suffer from malnutrition, as olfaction contributes significantly, as I just said, to the sense of taste. So for these people, food may lose much of its appeal. Now, many conditions actually result in anosmia or rather hyponosmia, or hyposmia, I'm sorry. Hyposmia is reduced olfactory sensitivity. And so anything that blocks air from reaching the olfactory epithelium, like nasal polyps, a deviated or fractured septum, swollen respiratory epithelium from an infection or an allergic reaction, all of these cases, the anosmia or hyposmia is typically temporary, and olfaction will return once airflow is restored. However, permanent anosmia is very possible. Head injuries that damage those neural pathways can result in permanent anosmia. And remember, that olfactory nerve actually sits directly on top of that cribriform plate, and so, or passes through that cribriform plate. And so damage to that very sensitive bone is highly likely to affect an individual's sense of smell. And the neural degeneration from Alzheimer's or Parkinson's often impairs the individual's sense of smell just by degrading those neurons. In fact, research tends to suggest that decreased olfaction may actually be an early sign of Alzheimer's disease. Now, hyposmia now naturally develops with aging, simply because fewer olfactory neurons are going to be replaced by basal cells. And this process tends to occur gradually, but once an individual becomes the age 60, it becomes particularly noticeable. Now, there is a question here on the bottom: why could anosmia or hyposmia be problematic? Well, what did I just mention? I mentioned they could not sense the smell of smoke. They could if there's a fire, this individual could not smell the smoke in time to be able to get out of their house. This individual could have a gas leak in their home and not be able to smell the gas additive that triggers most of us to notice that rotten egg, that sulfur smell. So without these protective abilities, this individual is at a higher risk of death, really, of being unsafe. Now let's talk about gustation, the sense of taste. Really, the flavor of a food encompasses many different types of sensation. Most of what we consider taste is actually just the smell of a food when it reaches that olfactory epithelium. This is why many foods can lack flavor when the nasal cavities are blocked, like when you have a cold. Now, other sensations like food temperature and texture, these are detected by those general sensory neurons. Some foods just quote unquote taste better when they're warm, or the texture of a food may not be appealing in spite of a pleasant taste. A lot of people have this with coconut. They may not like the texture, it's very fibrous, but they like the taste. And then spicy foods, remember, spicy foods, like they use that capsaicin that actually stimulates receptors for pain and heat in the trigeminal nerve, and then you interpret that as the sensation of spicy or hot. But it is not actually temperature hot, it is just activating that chemoreceptor. Now, A large part of our experience in eating a food does depend on the sense of gustation. This is the sense of taste. Like olfaction, gustation is going to involve chemoreceptors that respond to very specific chemicals. And taste begins with the stimulation of specialized receptor cells called taste buds. These specialized receptor cells are in the taste buds. And these taste buds are small clusters of these receptor cells with supporting cells scattered, and they're all across the tongue and parts of the oral cavity. Now, each taste bud is contacted by sensory neurons that transmit that stimulus to the central nervous system. So remember, this is a special sense. So these taste buds are your special sensory cell that transmit the signal to a neuron that goes to the CNS. And we're going to talk more about how taste sensation goes from detection of the chemical on the tongue to the processing of that neural signal in various regions of the brain. Now, your tongue is covered with rounded projections called papillae, or one of those would be a papilla. Now, papillae are classified into 4 groups based on their shapes. There are vallet papillae, otherwise known as circumvallet papillae, and these are the largest dome-shaped papillae, and each of these contains hundreds of taste buds. You also have fungiform papillae, and these are mushroom-shaped, getting their name, fungi, mushroom, and each of these only contains a few taste buds. You have foliate papillae that are ridges on the sides of the tongue and contain taste buds only when you're a child. And finally, you have filiform papillae that are long, thin cylinders scattered all across the tongue. Now, these do not contain taste buds, but do have texture and temperature sensing nerve endings. And here you have a great image out of your book, the anatomy of the tongue and the taste buds. So you see various papillae here throughout the image. You have fungiform papillae that are shaped like mushrooms. You have vallet papillae, which you see is zoomed in right here, containing those taste buds, which would be located here. So these are those specialized sensory cells, which then send a signal through the sensory neuron. And if we zoom in even more, you have the structure of a particular taste bud right here. And these generally contain 3 types of cells. They have gustatory cells. These are taste cells. These are the specialized epithelial cells with microvilli that contain receptors detecting different taste chemicals. These microvilli are found on the apical surface of the cell and project into a small pocket on the surface of the papilla known as a taste pore. So these receptors puncture through into a taste pore, and the basal end of each gustatory cell forms a synapse with the receptive endings of a sensory neuron. And so these sensory neurons then carry taste stimuli to the CNS through either the facial nerve, the glossopharyngeal nerve, or the vagus nerve. So those are cranial nerves 7, 9, or 10. And typically a taste bud is going to have approximately 100 of these gustatory cells. In addition to the gustatory cells, a taste bud will contain basal cells. You see these right here, basal cells. These are going to be those stem cells that differentiate into new gustatory cells. These are exposed even more than the olfactory cells. And so what happens here is these have a lifespan of about 10 to 14 days. So they're replaced every 10 to 14 days. And then you'll have the supporting cells right here. And these supporting cells surround gustatory cells. And basically they have physical support. They provide physical support for the gustatory cells, but they do not do anything for actually taste sensation. So as I said, yes, gustatory cells, basal cells, supporting cells in the taste buds. This just puts all of that in text so you guys don't have to keep pausing and re-listening to that presentation. Now, like I said, just like olfaction, this taste sense involves transducing chemicals. In this case, it's going to be chemicals from the food that we eat into electrical signals. And this transduction from chemical to electrical takes place in those gustatory cells, and the action potential is then transmitted to the CNS through one of those 3 cranial nerves. To understand this process, let's look at the chemicals we perceive as tastes. Sweet tastes are typically due to simple sugars like glucose or fructose. There is ethylene glycol and lead actually can also taste sweet, which is why there may be a problem with young children ingesting these substances, leading to accidental poisonings. You have sour tastes produced by hydrogen ions, and most of these are found in citric acid, like lemon juice. Salty foods. This salty taste is due to the presence of metal ions like sodium and potassium. Then you have bitter flavors. These are imparted through many different compounds like alkaloids found in coffee, plant toxins, and rancid foods. Now, humans are most sensitive to bitter tastes, which serves a sort of protective function, as a large number of bitter substances are actually toxic. Finally, you have umami, and this is the Japanese word for savory or delicious, and this is a taste often associated with meat or broth. This is that very savory taste, and it is produced by glutamate or other amino acids. Now, when we detect taste, that stimulus binds to receptors on the microvilli of gustatory cells. These receptors are classified by the substance to which they respond, and only one type of receptor is typically found on a particular gustatory cell. So that really means that despite popular belief, different tastes are not localized to different regions of the tongue. I know you guys have seen that tongue map where it shows you have, oh, this is your sweet side, and you have 2 salty sides. That's not accurate. And now, obviously, the foods that we eat are going to contain more of the flavors than the 5 mentioned here. And what happens here is each food's unique flavor lies in its particular combination of each of these taste chemicals, as well as the unique combination of taste receptors that they activate. So, like an example, a pretzel is going to taste differently from a potato chip, even though both are very salty. Now let's actually talk about how a taste receptor actually senses that taste. So first, a substance has to actually reach those taste buds in those papillae. And so this is really accomplished with the help of saliva. And you can actually you can test this yourself. Take a paper towel, dry off part of your tongue, and then pour a little bit of salt on it. You're not going to taste it as easily as if your tongue was naturally as moist as it's supposed to be. Your sense of taste is greatly diminished without that saliva. Now, when the taste stimulus does reach a gustatory cell, it has to be transduced into an electrical signal. And this is going to happen in 3 steps, essentially. First, those changes in ion movements are going to depolarize that gustatory cell's plasma membrane. So what actually happens here is different ion movements actually occur depending on which taste is actually being sensed. Sodium ions in salty foods and hydrogen ions in sour foods are going to go directly through their ion channel receptors and work to depolarize the cell. But receptors for bitter, sweet, and umami compounds have to activate a G protein. And this G protein indirectly closes potassium ion leak channels. And so this decreased movement of potassium ions out of the cell produces that gradual depolarization. So what do we have now? We have the depolarization of the membrane opening those voltage-gated calcium ion channels, and now calcium ions are going to enter the cells. Okay, now unlike a neuron, this gustatory cell does not actually generate an action potential. Gustatory cells are small enough that the local potentials generated here can quickly spread across their membrane, making an action potential, a long-reaching action potential, unnecessary. And so what do we have next? We have the calcium ions triggering the release of neurotransmitters, which produce an action potential in the axon of the sensory neuron. And so these gustatory cells release these neurotransmitters. These neurotransmitters then bind to the axons that they're close to, and these can include serotonin, they can include glutamate, acetylcholine, and GABA. And so once they bind to those ligand-gated voltage channels, then they'll start generating local potentials that summate and become action potentials, traveling through one of those three cranial nerves, being interpreted as taste. Here you can see exactly what I'm talking about. So salty and sour, those sodium and hydrogen ions go straight in through those ion channels. Well, if they're not salty or salty or sour, they have to activate that G protein, and then the G protein is going to close potassium. Let's say there's a potassium leak channel right here. It's going to close those potassium leak channels, leading to a gradual depolarization. Okay, so once they're depolarized, that is then going to activate these calcium ion channels, and the calcium is going to flood in. Okay, and once that happens, then you have that local potential. And like I said, these do not require action potentials. They're very small, so these local potentials are actually perfectly fine traveling down and generating these neurotransmitters. So once those action potentials are triggered, the local potentials trigger the release of these neurotransmitters. Okay, then the neurotransmitters are going to bind to their ligand-gated ion channels here in this sensory neuron, and they're going to create an action potential that goes into the CNS. I'm sorry, I skipped through the presentation a little bit there. Now remember, axons of the facial, glossopharyngeal, and vagus nerves carry the stimulus directly from the tongue into the CNS. And the facial nerve is going to transmit taste stimuli from the front 2/3 of the tongue. The glossopharyngeal transmits taste from the rear third of the tongue, while the vagus nerve has a minor role in taste that basically innervates various taste buds that are scattered along the base of the tongue, and then some on the posterior palate. So let's see, the axons of these three nerves are going to terminate in what is known as the solitary nucleus of the medulla oblongata. Okay, they synapse from there onto central sensory neurons. This solitary nucleus is essentially a cluster of somas located in one of the columns of the medulla oblongata, where the axons transmitting taste signal will terminate. And some neurons in this solitary nucleus connect with parasympathetic neurons of the facial and glossopharyngeal nerve, and these work together to control that reflexive production of saliva. Now, once axons from the solitary nucleus synapse onto neurons in the thalamus, okay, This then sends the taste signals to the primary gustatory cortex in that parietal lobe, or in the insula. And the primary gustatory cortex is going to be responsible for integrating all of that incoming stimuli. Well, that leads to an awareness, especially an identification of a particular taste. Now, once the insula integrates that signal, they may also be relayed to the inferior part of the frontal lobe. And this is sort of where they're integrated with visual and olfactory stimuli to form a quote-unquote complete picture of what you're eating. And so to your limbic system, this produces an emotional reaction to taste, either pleasant or unpleasant. So yeah, axons of each of these cranial nerves is going to carry signal from the taste buds up into the solitary nucleus in the medulla, okay? Then the medulla is going to transmit that signal to the thalamus, and then the thalamus is going to send that signal to the primary gustatory cortex, okay? And then from the primary gustatory cortex, it may also travel to the insula. So that is where all of that integration occurs. So this is just that pathway from taste bud, from actually receiving that chemical sensory signal and transmitting it out into the CNS. And remember, it's either cranial nerve 7, 9, or 10, depending on which region of the tongue is being used. And remember, all of these different regions have different have taste buds within them. So, and each taste bud itself has various gustatory cells which are responsible for each of the individual tastes. So remember, that's not, you're not divided into taste buds that look like this, okay? That's wrong, okay? So what you actually have are those spread throughout all over, unless you're a child, in which case you have these particular structures on your tongue. And again, those should fade as you age into an adult.
Now, this will begin pretty much the longest subsection of our chapter this week. This is going to be the anatomy of the eye. And so if we talk about the anatomy of the eye, first we're going to talk about the anatomy of the eyeball itself. And then later on in this chapter, we'll talk about the anatomy of the eye's accessory structures. So the first thing is the eyeball. And the eyeball is essentially a hollow sphere, approximately 1 inch in diameter, resting in the anterior portion of each orbit of the skull. And each one basically only occupies a third of the orbit, with the remainder of that space being filled with fatty tissue that cushions the eye and holds it in position. The eyeball consists of an outer wall that encloses several inner chambers, and then it also consists of the lens, which focuses light as it enters the eye. And we'll talk about that more as we keep going through here. The eyeball itself has many layers, 3 of them to be exact. It has the fibrous layer, the vascular layer, and the neural layer. And each of these has its own set of functions, including maintaining the shape of the eyeball and then transducing light into action potentials. And this is your basic structure. I'm going to show you this now so you have a general idea of what I'm referring to. And then at the end of this discussion of layers, I'll show this image to you again so that you know roughly to what I'm referring. So yeah, there's the fibrous layer right here, consisting of this white outer area, this cornea, this sclera, right, is this white area, and then this exterior most area that protrudes it and through which light actually travels. That is the cornea. So the sclera is the white of the eye, and the cornea is this outer covering just in front. Then you have the vascular layer, and the vascular layer is going to consist of the iris, which is the colored portion of your eye, the ciliary body, which is this sort of flowery looking structure, and as well as the choroid. the choroid layer is this very vascular layer here. You see all of the blood vessels traveling through it. You can see veins here and arteries here supplying this eyeball with blood. And then what you have is the neural layer. And the important thing about the neural layer is this is where the retina is located. And this is where all of your photoreceptors are located. And it's this interior-most layer, this deep-most layer, where all of that transduction of light into action potentials actually occurs. So again, I'm showing you this now, and I'll show it to you again later so that you know exactly where everything is. Now, as I said, the fibrous layer is essentially the outermost layer of the eyeball. It consists of both the sclera and the cornea. The sclera is going to be the quote-unquote white of the eyeball, and it covers about 5/6 of the surface of the eye. The sclera is essentially white due to its collagen content, and this collagen content is what allows it to resist that deformation from external and internal forces, maintaining its shape. The white color is also due to the irregular arrangement of these collagen fibers, making that sclera white or opaque. Then the cornea is actually continuous with the sclera, and where the sclera is white, the cornea is relatively translucent. And this covers the anterior-most 1/6 of the eyeball. And this allows that the parallel arrangement of that collagen allows light to pass through. And that is the most important part here, because that plays a huge role in admitting light into the eye, focusing it, and then it also has a great example of structure function, which you'll call back to from A&P 1. Now, the cornea is actually avascular. There are no blood vessels in the cornea, so it actually obtains all of the oxygen it needs from the fluid behind it, as well as from the air. People that wear contact lenses that sit directly on the cornea, well, these are actually designed to allow oxygen in and reach your cornea. There are extended wear lenses that can be worn overnight, and these are specifically designed to have larger oxygen pores, and that reduces corneal swelling as well as a reduction in the risk of infection. Now, the cornea also contains many nerve endings that convey pain, but very few that actually relay touch, and that is why we essentially can tolerate smooth contact lenses, but not jagged particles of dust. And here is again that image with the white collagen in the sclera, and then you see it runs sort of parallel. Those collagen fibers look more like this, and they allow the light to pass through in that manner. And these are all sort of crisscrossed, not allowing light through. I hope that makes sense. Now, the vascular layer, it is located immediately deep to the fibrous layer, and it has those three parts. There's the choroid, and this is the most extensive component of the vascular layer. This is richly supplied with blood vessels, as its name implies. And these blood vessels are vital because they supply oxygen and nutrients to the neural layer of the eyeball. The choroid layer also contains melanocytes, which produce a pigment, and that pigment actually reduces the scattering of light in the eye. The reflection of light from a camera's flash, you'll see that red eye effect. That is actually due to the large amount of blood in the choroid. The other part you have is the ciliary body, and this is actually continuous in the front of the eye with the choroid. And this is essentially a ring of smooth muscle that surrounds the lens. You can see where they meet at the scalloped edge of this ciliary body, and this structure is known as that ora serrata. That ora serrata is that flower-like structure that I showed you. And then there you're going to find these fine threads called suspensory ligaments. And these suspensory ligaments actually connect the ciliary body to the lens. And that through contraction and relaxation of smooth muscle, they change the shape of the eye. And that changing of the shape allows near and far vision. You also have the iris in this section, and the iris is just an extension of the vascular layer that is just in front of, that is just anterior to the ciliary body. Now, this is that visibly pigmented portion. You know, that if you have brown eyes, blue eyes, green eyes, this is where you're going to see that color. And all of that color surrounds your circular pupil. And the pupil is not actually a structure. The pupil is the absence of a structure. It is a hole in the iris. And this is where light actually enters the eye. And the iris contains smooth muscles, 2 smooth muscles to be exact, that constrict and control how much light passes through the pupil. The first is called the pupillary sphincter muscle. And this constricts the pupil, reducing the amount of light entering the eye, and that comes from parasympathetic nervous system stimulation. Remember, which goes more with the pupillary dilator muscle. Okay, this is the second one. The pupillary dilator muscle has fibers arranged around the pupil in a radial-like pattern, a star-like pattern. And these fibers are stimulated by the sympathetic nervous system because what they're doing is they're dilating your pupil. So you're letting more light in. Remember when your fight or flight system kicks in. And if you've ever had your eyes dilated during an eye exam, what this actually is that those drops they put in your eye are actually blocking your parasympathetic activity of those smooth muscles. And so that allows the sympathetic activity to overcompensate and dilate those pupils without any opposition. Now let's look at this, okay? We're looking at the vascular layer here. Well, we got the iris, that melanin concentrated colored section, right? Well, under that, what do we have? We have the ciliary body, this structure here, and we have those suspensory ligaments here connecting to the lens. We have the ora serrata here, this flower-shaped structure. And then all of this middle layer you see here with all of these blood vessels, that's the choroid, okay? What else? You see those, you can see a cross section of those smooth muscles here, the pupillary dilator and the pupillary sphincter muscles. Obviously, this empty area here would be the pupil. And that's about it for the vascular layer. So this shows you exactly what I was referring to when I said how the pupil dilates and constricts. So under parasympathetic control, right, these pupillary sphincter muscles are going to constrict. They're actually going to constrict and make your pupil smaller. Okay, let's say this is normal. This is our baseline here. Well, when the parasympathetic control actually kicks in, it's going to sphincter, it's going to close that sphincter and constrict the pupil. Then, let's say we're in an emergency situation or you're anxious about the upcoming test. Well, you're going to actually dilate your pupils, and that's through the sympathetic system again, remember. And what happens there is the pupillary dilator muscle contracts and it opens up the pupil and makes it much more open for light. And these arrows on the top, I want you to remember, are referring to, let's say you're it's noon here in this photo, right? And then this one would be at dusk. So the intensity of the sunlight, the intensity of the light of your environment is different. It's much higher at noon, right? So your pupil is going to constrict, reducing the amount of light that actually gets in there. But at night, you need more light in there. So what you see here is that decreased light intensity of your environment, and that allows more light in through your pupil, allowing you a greater chance of being able to see a potential threat. Now, the deepest layer of the eye is the neural layer. But we're probably just going to skip calling it the neural layer and use its common name, the retina, because that is what most commonly it is known as. And it has two layers. The superficial layer is very thin and pigmented epithelium that reduces the scattering of light. Then deep to this, you're going to find a layer containing photoreceptor cells, and these are known as rods and cones, and each of these has a specific function. Rods are photoreceptors that are responsible for black and white vision in low light levels, and also for peripheral vision, okay? Whereas cones are responsible for high acuity color vision in higher light levels. An easier way to remember this is cones has a CO, so does color vision. That's how I remember it. Now, the retina's epithelium and the photoreceptors are actually nourished by those choroid blood vessels, while deeper neural structures of the retina are supplied by branches of the central retinal artery, and this is going to enter the eye through the optic nerve. Typically, during an eye exam, you'll be able to see these structures. Now, at the central posterior retina, you're going to find a yellowish area known as the macula lutea. This is the yellow spot, and this contains a high concentration of photoreceptors. And I want you to note that in the photo I'm about to show you, the macula lutea does not appear yellow. And this is going to simply be due to the red eye effect from the light used to view the retina. So it's just that reflection off of the choroid. In the center of the macula lutea is an indented area called the fovea centralis. Fovea just means pit. So this is the central pit of the eye. And in this, you're going to find cones that are very tightly packed together. And so this high density of cones actually makes the fovea centralis this high definition area of the retina. And it allows for extremely detailed vision as we focus on a particular object. Something I forgot to mention in that last slide, the importance of that macula lutea is extremely important for high acuity vision. And so as you age, your vision is gonna worsen. Okay, this is just an age-related side effect, and it can come along with a condition called macular degeneration. So macular degeneration is essentially a progressive loss of visual acuity, and so this is where people may require reading glasses as they get older, or they may have difficulty with color perception. Now, the axons of the optic nerve, okay, the axons of these nerves are going to gather at what is known as the optic disc. And this optic disc is actually a portion of the retina that does not contain any photoreceptors. And so for this reason, this area is actually not capturing any visual image. And so it is called the blind spot. And we don't typically notice a blind spot because your brain is very good at filling the image in. There is a test image I can show you that you'll be able to pick up your laptop or your computer or get closer to your monitor, and as you get close, you notice that the empty spot in an image will disappear. That is you finding the distance for your blind spot. Now, the two layers of the retina are usually in contact with each other, but they are not physically linked by any desmosomes or any tight junctions. So for this reason, certain conditions, especially trauma, diabetes, or if a person just has an abnormally shaped eyeball, they can actually separate the inner layer away from the pigmented epithelium. And this is a condition commonly known as a detached retina. And this isolates the photoreceptors from their blood cells or their blood supply. And so this is a very, very serious condition. Permanent loss of vision can actually occur if it is not corrected within several days. And here you have that photo of the interior of the eye. Remember, I said this was not going to appear yellow because of the, this area was not going to appear yellow because of the reflection of the light off the choroid. Now, it is quote unquote actually yellow, and I don't want you to confuse it with this area, the optic disc. But here you do see that optic disc, and this is where the axons of the optic nerves are going to coincide. And I really don't want you to confuse the optic disc with the fovea centralis. Okay, so because the difference there is massive. Okay, here we have lots and lots and lots and lots of cones giving us high definition vision. Here we have no rods, no cones, no visual signal is being interpreted there. So that is the blind spot. Blind spot here, I'm going to put a B there, and then HD vision here. I hope that it's not Harry Potter, I promise, it's HD. I hope that will make it a little bit simpler for you to notice. Or to study, at least. And so this is that image I was talking about. So if you want, you can pause it and follow the directions. And as you get closer, you'll either notice the dot will disappear, or as you get closer to the X on the bottom, you'll notice that the pencil actually appears connected. And so that is you actually putting that visual information directly in front of the blind spot. And so your brain is just connecting that image. And so what is the lens? Well, for our purposes, we're going to need to know that the lens is a slightly flattened sphere, and this is sitting just behind the pupil and the iris. And the lens's entire job is going to be directing light toward the retina from nearby objects. Now, this lens is connected to the ciliary body, and remember, the ciliary body is part of the choroid layer. And this is actually connected to the lens via suspensory ligaments, and those control how round or how flat the lens is, giving us that near or far vision. The lens also contains a few cells called lens fibers, and these essentially lack nuclei, and they're very tightly packed, giving us that transparent or translucent even, color. And here is where we're going to see that structure right here. Now this rounded, flattened disc is the lens, and it is connected through these suspensory ligaments here to the ciliary body here. Oh, goodness, excuse me. So this lens's entire job is going to be taking parallel light rays from the reflected from the environment. And what it's going to do is it's going to take those parallel light rays and it's going to redirect them to this fovea centralis. Remember that high definition vision area. That's where we want to be. So it's going to take those light rays and direct all of them up into the fovea centralis. Now, what happens if the lens is not clear? Well, then you're going to suffer from a condition known as cataracts. And this is essentially vision being impaired despite the fact that functional photoreceptors are still present. So a clouded lens or a cataract is one of this is one of the most frequent causes of blindness. And causes include trauma, UV exposure, and then diseases like diabetes can also promote cataract formation. But one of the most common causes is just aging. And as lens fibers age, they tend to progressively get darker and less organized. And so that turns this lens from transparent or translucent to a milky white. And a cataract cannot be reversed, sadly. So what happens here is a surgeon will remove the lens, the natural lens, and replace it with a synthetic one. And this typically restores vision, although for most patients, glasses or contacts are needed for minor adjustments. Now, the eyeball isn't hard all the way through like a baseball or something. What it has is it has cavities and chambers, okay? So let's talk about the cavities first. This largest cavity is the one in the back, behind the lens. This is the posterior cavity, and this is filled with a gelatinous material, mostly 99% water, but it has collagen fibers, hyaluronic acid, and a few cells on the periphery. This is called the vitreous humor or vitreous body. And this jelly fluid is actually the source of some of those floaters, if you've ever experienced one of those. What this actually is cellular debris trapped in the vitreous humor that casts a shadow onto your retina. And now floaters increase with age, but that's just because the vitreous humor tends to shrink and is not replaced. Please also note that you're going to see near or passing through this cavity is a small canal, the hyaloid canal. This passes through the vitreous humor from the lens to the optic nerve. And then after birth, this essentially contains fluid that assists in changing the shape of the lens. Now, that's the posterior cavity. The anterior cavity is found in front of the lens and the ciliary body. And the anterior cavity is divided into two chambers. The anterior chamber in front of the iris, between the iris and the cornea, and the posterior chamber between the lens and the iris, okay? Two chambers of the same anterior cavity. And these chambers are filled with a very watery fluid known as the aqueous humor, where the vitreous humor is very gelatinous with all that collagen. The aqueous humor is very much watery. And this essentially flows from the posterior chamber through the pupil into the anterior chamber. But this has to drain at essentially the same rate that it is created. And this is done through a blood vessel that circles the anterior ridge of the iris called the scleral venous sinus. What happens if it doesn't drain? Well, then you're going to get a lot of pressure in that area, and that may lead to, you may know that more as its common name, glaucoma. And I'm sorry, this is the image here for this, this eye. So we see everything, let's say we'll just divide the eye, let's just say we divide the eye like this. And then this is the posterior chamber, the posterior cavity. And this is the anterior cavity. Oh God. Please don't judge my mouse handwriting. Okay, so this is the anterior cavity, that's the posterior cavity. Well, now the anterior cavity is divided again, basically like this, to where we see posterior chamber and the anterior chamber. Oh God. Yeah, I just noticed these were here. Okay, so yeah, it's the same thing. Posterior chamber behind the iris and behind the pupil, essentially this area here, posterior chamber, and then anterior chamber here. And those are part of what? The anterior cavity. Oh God, now you can't even read that. And then this is the posterior cavity. And then what is this again? This is that hyaloid canal, and that's filled with fluid that helps the posterior cavity I'm sorry, it's filled with fluid that changes the shape of the lens. Now, if aqueous humor doesn't drain and that fluid builds up, what it's going to do is it's going to raise the pressure inside the eye in what is known as intraocular pressure. This elevated pressure pushes on the vitreous humor, and then that pushes on the retina, and that can damage it and the optic nerve. And so glaucoma, and this is the second leading cause of blindness, glaucoma can actually result from eye infections, medications, congenital defects in that venous sinus, and then most people that have glaucoma actually don't know, I'm sorry, don't have any other symptoms aside from gradual loss of vision. And this can actually occur so slowly that it's not noticed until the disease has become very advanced. And lost vision is not restorable, but progression can be stopped or slowed with medications. These medications either improve the drainage, they reduce the amount of aqueous humor produced. And then if medications fail, what can happen is surgical intervention. Someone will stick a needle into this cavity, this anterior cavity, and they will aspirate the fluid out, and that will relieve the pressure. Now that we've talked all about the eyeball, let's talk about the accessory structures of the eye. And these are going to be your eyelids, eyebrows, eyelashes, conjunctiva, lacrimal apparatus, and the extrinsic eye muscles. And all of these have roles in either protecting the eye or moving the eyeball. First, let's start with the eyelids, otherwise known as palpebrae. These are two thin folds that cover the frontmost portion of the orbit. Their basic job is to prevent things from getting in. They also distribute tears across the surface of the eye during blinking. And these eyelids, they're not loose or anything. They're actually stiffened by what is known as a tarsal plate. This tarsal plate is a thin piece of dense, regular connective tissue. And then inside of that tarsal plate, you're going to find modified sebaceous glands that are called tarsal glands. And these work together to secrete oil that prevents the eyelids from sticking together. Now, the superior, the upper, and the inferior, the lower, palpebrae, the upper and lower eyelids, meet at the edges of the orbit known as the medial and lateral commissures, also known as the medial and lateral canthi. Now, in the medial canthi, you're going to find the lacrimal caruncle, which is that fleshy pink structure found at that medial commissure, and that secretes, it contains sebaceous glands that secrete a very whitish lubricating substance. You also find two skeletal muscles moving the eyelids. Both the superior and inferior eyelids contain portions of the orbicularis oris. The orbicularis oculi muscle. The orbicularis oris is around your mouth. The orbicularis oculi is around your eye. And these allow you to tightly close your eyelids. And then the levator palpebrae superioris muscle actually inserts into the superior palpebra and elevates it. So when you open your eyes really wide, what you're actually doing is you're opening your upper eyelid more with that levator palpebrae superioris. And we should see that in the next photo. So let's go through those structures. Superior palpebra, upper eyelid, right? Aims up right here. And inside of that, you're going to find, oh my gosh, this is the tarsal plate. And then down inside the tarsal plate, you're going to find all these glands, those sebaceous glands. Those are the tarsal glands. Above it, you're going to find, oh, what's this? This is the orbicularis oculi muscle. And then back here, you have that levator palpebrae superioris muscle. So this is going to draw the eyelid up and back. This one's going to allow the eyelids to squint closed. What else we got? Lower eyelid, the inferior palpebra. Remember, palpebra is the singular form of palpebrae. What else? We'll get more into some of this, but the lower eyelid also has the tarsal plate and the tarsal glands and the orbicularis oculi muscle. Now you have the eyebrows and the eyelashes, okay? And these serve protective functions. Any hair really serves a protective function. Hairs along the ridge of the brow form the eyebrows, and these eyebrows actually prevent perspiration from running down straight into the eyes. They also work to reduce glare in bright light, and they form important facial expressions. The eyelashes are stiff hairs located in the edge of the superior and inferior polypary. They are richly innervated by nerve endings so that the slightest touch will trigger that blink reflex, and that reduces the risk of eye injury. So as you know, like something's coming in to touch your eye, you're going to blink, and when you blink, your eyelids are covering your cornea, preventing damage to it. Then you have the conjunctiva. And what is the conjunctiva? Well, it's a thin epithelial membrane lining the posterior surfaces of the eyelids. These are the palpebral conjunctiva, and then it turns back on itself as the bulbar conjunctiva. And this covers the anterior surface of the white part of the eye. This conjunctiva is translucent, and that allows you to see tiny blood vessels beneath it. You may have gotten pink eye. This is a viral or bacterial infection producing inflammation in the conjunctiva. This is conjunctivitis, and it is typically highly contagious. The lacrimal apparatus is what's going to produce tears and then also drain them from the eye. So first, there's the lacrimal gland, located in the superolateral region. So where is that? It's up and on the outside of the eye. So just posterior to the conjunctiva is where that is located. When this is stimulated by autonomic neurons, tiny ducts release tears and mucus into the conjunctival sac, lubricating the eye and washing away dust and debris. Well, where does it go after that? Well, blinking is going to sweep the tears medially and inferiorly across the surface of the eye, where they then drain into a series of passages that lead into the nasal cavity. This is these tears first enter the lacrimal puncta, or if it's just one, it's the lacrimal punctum. And these are tiny holes in the medial edge of each eyelid. These puncta are continuous with the lacrimal canaliculi, or lacrimal canals, which are small ducts emptying into the lacrimal sac. This is located in a small depression in the lacrimal bone. This lacrimal sac drains into the nasolacrimal duct, and then that is where that actually travels through the lacrimal and maxillary bones, reaching the inferior nasal meatus. And this is, there's a photo here that'll show you exactly how narrow these ducts get through an X-ray. If you produce tears in response to debris, allergens, or emotions, you'll probably end up with a runny nose, right? And excess tears may also spill over onto your cheek. The connection between your nasal cavity and your eye can also be a two-way street. Pathogens can easily travel from your nose to your eye, resulting in conjunctivitis or a painful infection of the nasolacrimal duct. It also explains why some people, when they're drinking water or they get water in their nose, they may eject liquid from their eyes. They're not actually doing it from their eyes. They're doing it through this lacrimal system. So here you see that X-ray I was talking about. We see how narrow these ducts actually get. And so these are the entrances here that you see represented here. The lacrimal canaliculi, But tears are produced in the lacrimal gland, and then they sweep across and get drained through one of these canaliculi down into the lacrimal sac. The lacrimal sac then goes down through the bone and into that inferior nasal concha, that inferior nasal meatus, draining out through your nose. So your runny nose is actually just more tears. Now we have the extrinsic eye muscles. All right, so if you think about your eyes without muscles, what are you going to do? Well, every time you got to look at something new, you got to move your neck. But because we have a set of extrinsic eye muscles, we're able to move our eyes up, down, medial, laterally. And there are 6 of these, 6 skeletal muscles that insert into the outer layer of each eyeball. And each extrinsic eye muscle actually originates from a common tendinous ring on the posterior wall of the orbit. And these muscles have names. There are 6 of them, and their names are the superior rectus muscle, the inferior rectus muscle. There's the medial rectus muscle, the lateral rectus muscle, and then you have the oblique muscles, both superior and inferior. Let's first talk about the superior and inferior rectus muscles. Well, both of these insert into the superior and inferior portions of the eye and move the eyeball superior and inferiorly. However, their origin lies slightly medial to the eyeball, so both muscles are able to move it medially as well. Excuse me. So the superior rectus muscle moves the eyeball up and medially, superiorly and medially. The inferior rectus muscle moves it inferiorly and medially. The medial and lateral rectus muscles insert into the medial and lateral aspects of the eyeball, respectively, and move it medially and laterally, respectively. Medial moves it medially, lateral moves it laterally. Then you have the oblique muscles. The superior oblique muscle travels from the posterior orbit along that medial wall, and then it travels through a fibrous loop called the trochlea. And this trochlea is just that fibrous loop. It's sort of an anchoring point before this superior oblique muscle will then insert onto the superolateral part of the eye. And it depresses the eye and draws it laterally. And when it works together with the inferior rectus muscle, it moves the eye directly inferiorly. Okay, that's the superior oblique. Then you have the inferior oblique. This is going to sweep from the medial floor of the orbit to insert on the inferolateral part of the eyeball. And this inferior oblique elevating the eye, this inferior oblique elevates the eye and moves it laterally. There's a figure that we're going to show here in just a little bit that'll maybe clear all this up for you. Now, these muscles also produce these muscles must produce very small, precise movements, and so they are one of the most highly innervated skeletal muscle groups in the body. Some of the motor units consist of only 2 or 3 muscle fibers compared to 1,000 in muscles of the leg. And so 3 of the cranial nerves, the oculomotor nerve, the trochlear nerve, and the abducens nerve all work together to innervate these extrinsic eye muscles. Oh, I'm sorry. So that's what this figure says. So yes, oculomotor innervates the eyes, the extrinsic eye muscles. Trochlear does it as well, and so does the abducens. Injury to any of these cranial nerves can weaken or paralyze eye muscles. If eyes can't move together, they send slightly different images to the brain, and this is actually interpreted as blurred or double vision, known as diplopia. This is a condition known as diplopia, and the truth is that if diplopia is present at birth, this child is said to have a quote-unquote lazy eye or a strabismus. Strabismus can sometimes be corrected by covering the good eye with a patch, and that will force the use of the affected eye, and that will strengthen those weak muscles. If it is not corrected in time, these muscles will atrophy and diplopia will become permanent. So a thing that students can use to study this is to remember which nerves innervate which extrinsic eye muscles is LR6, SO4, all the rest are 3. Lateral rectus is done by cranial nerve 6. Superior oblique is done by cranial nerve 4. All the rest are 3. So lateral rectus 6, superior oblique 4, all the rest are 3. That should make sense for you guys. And then if you remember O to touch and feel, very green velvet, heaven. You should be able to remember 4, 6, and especially 3. Olfactory, optic, oculomotor, trochlear, trigeminal, abducens, facial, vagus, glossopharyngeal, vestibulocochlear, glossopharyngeal, vagus, accessory, hypoglossal. You guys should remember that. Well, we've talked all about the actual structure of the eye, but how do you see? That's what we're going to talk about now. And to understand how you see, first we have to talk about the principles of light itself. So, in the chemistry chapter, you guys should have discussed how there's different forms of energy, and energy is defined as the capacity to do work. Well, one of these forms of energy is electromagnetic radiation, and this in certain wavelengths is measured in nanometers, and this is visible as light in a specific range. These range from very short, very powerful wavelengths to those like gamma rays and X-rays, to much longer, lower power wavelengths like microwaves and radio waves. Now, these are all invisible light, quote-unquote, invisible light frequencies. The visible light frequency is the range of wavelengths that we can detect with our eyes. We're able to perceive segments of that range as particular colors. Shorter wavelengths, about 420 nanometers, are like blues and violets, with longer wavelengths, like 660 nanometers, are more red. And then regardless of wavelength, light as a unit is measured as a photon. And a photon is what is going to stimulate the photoreceptors in the retina. This is what I mean when I say electromagnetic energy. Well, it is on a spectrum, and I'm going to use, let's say, just black for this. So these very, very short energy waves over here are invisible light. And here you have gamma radiation, you have X-ray radiation, and then you have ultraviolet radiation, okay? And then on the opposite end, you have very long wavelengths, okay? And these are infrared, microwaves, and radio waves. Well, visible light is right in that sweet spot from about 350 to 750, and it goes, you guys know, Roy G. Biv. Well, that actually goes backwards, like this. That is a descending order of wavelength, where red is the longest waves. So red, orange, yellow, green, blue. We'll say indigo is there, and violet. So Roy G. Biv actually just works backwards on this one. And you guys aren't going to need to know specific nanometers, but do know 350 to 750 is the visible light spectrum. Now, rays of light actually bend in a process called refraction. They bend when they pass through certain translucent objects, and the amount of refraction that an object will provide is called its refractive index. Air typically has a refractive index of 1.0, meaning that light passing through the air is not significantly bent. If something has a refractive index higher than that, like water, light is refracted. You've seen this if you've ever stuck a straw in a glass and you notice that there's a disconnect where you're sticking the straw in the water does not match up with where you see the straw in the water if you're looking at the glass from the side. This is due to the air having that higher refractive index than the water having a higher refractive index than air, so the light rays entering the water refract. And I'll show you an image here that'll clear that up. But the degree to which light refracts also depends on the angle at which light is striking that surface. The greater the angle, the greater the refraction. And this has particular consequences for curved surfaces, as they are more angled at the edges. And so light hitting the edge of a curved surface is bent more than light hitting its middle. And if you've ever used a magnifying glass to burn ants, you know exactly what I'm talking about. Those light rays are going to converge, move inwards to a central point. This when the rays converge on one point, this is called a focal point, and they are said to be focused. And this is generally done through a convex lens. A concave lens has a surface that caves in, okay? So a convex has a convex surface looks like a magnifying glass. It curves outward. A concave lens curves inward. And what it does is it actually causes light hitting the edges of the lens to bend outward or diverge, so they do not actually focus. So here you see that image that I was talking about. You see air passing, or light passing through air, no refraction. But let's say you've stuck that straw in the water. Well, now all of a sudden you're going to see it like over here, right? That's because that light refracts more. The refractive index is higher than that of the air. So you see the light refracting. And then you have convex versus concave. You see this is curved inward for convex and then curved outward for concave. And so this would be the example there of an ant being burned by a magnifying glass. This poor ant was just going about its day, and then we all just burned it up. Everybody was a kid once. And then you have diverging rays that actually don't go to the center. They sort of scatter. Fun fact, most light crews, like for filming movies, and I'm sure you guys know YouTubers, they use lights with concave shrouds on them, and that scatters the light more evenly across their filming areas. But how does any of this relate to the eye? Okay, well, to provide clear vision, rays of light have to focus on the retina, okay, not behind the retina and not in front of it. Unfortunately, when a light will strike a non-transparent object like an apple, light rays are going to be reflected and scatter away from that object in a diverging pattern. Okay, we'll see a figure here in just a second, showing that when light strikes a non-transparent object, you're going to see light rays reflecting close to an object. They still diverge very sharply, okay? But if they're if they're reflected from an object from an object about 20 feet away, they're nearly parallel. Okay. In either case, the rays have to be refracted to converge and focus your retina. And this is where your cornea and your lens come into play. Okay, the greatest degree of refraction, about 2/3 of it, actually occurs as light passes from the air through the cornea. The cornea, remember, is filled with that aqueous humor, very liquid. It's very similar to that of water. And this can also explain why your vision underwater is very blurry. Light passing from the water to the cornea doesn't refract as much. And so it's very hard for that light to focus at the correct in the correct area. Some additional refraction occurs as light passes through the lens, but the refractive index of the lens is very similar to that of the aqueous humor, so the lens doesn't provide as much refraction as the cornea. For this reason, the lens typically provides fine refractive adjustments, okay? Now, as we just said, rays of light from distant objects are nearly parallel when they reach the eye, so they need little refraction to be focused on the retina. The cornea does most of the work here, and the lens stays in its normal flattened shape. This is the emmetropic state. This is the lens being flattened at rest, with the cornea providing most of the refraction. However, diverging rays from nearby objects, like the apple after you pick it up and look at it in your hand, these require a greater degree of refraction. So you see here, a great example here. You see light is still diverging. There's not enough space for those light rays to become parallel at ranges closer than 20 feet. Where at 20 feet you see they're basically parallel. That's why you're looking at the apple on a tree from 20 feet away. Your lens is in that emmetropic state, and then you have to accommodate when the lens is much closer. And what happens here is the lens actually changes its shape from flattened to round in a process called accommodation. So when this lens is now rounded, its curved surface refracts light to a greater extent, allowing it to focus on the retina. So this is that far vision versus near vision. So how does the lens actually change its shape? Well, the answer lies in those sphincter-like smooth muscles found in the ciliary body. When you're looking at distant objects in that emmetropic state, the smooth muscle of the ciliary body relaxes. This moves the ciliary body away from the lens and tightens the suspensory ligaments, flattening the lens, reducing refraction. When accommodation occurs, you're viewing a nearby object, ciliary muscle is going to contract, and the ciliary body moves closer to the lens. This is going to decrease that tension on those suspensory ligaments, which reduces their pull on the lens. And then without that tension, this lens is going to round up. It's going to become more round into an almost spherical shape. And this change in shape makes the lens more convex, increasing refraction and focusing the light from nearby objects onto the retina. But accommodation isn't the only event here to focus focus light from nearby objects. Two other things have to occur. Pupillary constriction, okay, has to occur. So light rays entering the edge of the lens cannot be refracted enough. And so that can actually produce a blurry image on the retina. So the pupils constrict slightly to cover the edge of the lens. And then this allows, let's say, dilation of the pupils during an eye exam is going to prevent that constriction. And that's why you can't focus as well on close objects after the exam until your pupils return to normal. Okay, so that's the first thing, in addition to accommodation. You also need convergence. So to focus near objects on the fovea of each eye, the eyeballs have to move medially. And so that movement of the eyeballs is actually called convergence. And so you can actually, if you look at someone when, like, let's say you have them look at the wall behind you, and then you say, okay, now focus on this pen, and you put the pen a foot in front of their face. You're going to see their eyes cross, and that they're going to move medially, and the pupils are going to constrict. And the lens is also going to change shape, but you're not going to be able to see that. So what people might not understand this, especially it was backwards to me. If you're relaxed, then why are the ciliary muscles tightened? Okay, why are those ligaments tightened if they're relaxed? Well, you're assuming that tension is a muscle contraction. All right, let's go back. Remember that these sphincters are circular muscles. Okay, when they are open, they are relaxed, and then when they're contracted, they're closed. So a relaxing circular muscle, like the ones in the ciliary body, are going to be open, and they're going to open up and tighten those ligaments that pulls the lens into a flattened shape. And then when they're contracted, they're going to shrink down, and the ligaments don't change in length, so they actually slack up, and they allow the lens to return to a more rounded shape. Let's see, there should be an image here. Perfect. So in the relaxed state, you see these muscles have contracted into a resting state, pulling these ligaments very taut, right? And because they're taut, they're pulling this lens into a more flattened shape, okay? And that's emmetropic. Remember, that's emmetropic for distant vision. Now, in the opposite side, let's say this muscle contracts, and you see it contracts, it moves in. These ligaments don't change length, so they actually slack up on this lens, and you see it becomes significantly more rounded. And that rounding is just like the magnifying glass. It's going to take those light rays that are coming in like this, and it's going to focus them just onto that macula lutea. So remember, constriction makes this loose, makes this round. Relaxation makes this tight, makes this flat. And this is the process of accommodation. And then remember, you're also going to have pupillary constriction and medial eye movement, known as what? As convergence. Now, you guys have seen me in the Teams meetings. You know that I'm wearing glasses. I've seen you guys in your tests. I know most of you guys wear glasses. This is actually going to be an error in refraction. We're wearing glasses because our lenses or our corneas are misshaped. And so there's a limited accommodation due to a misshapen lens or shape of the eyeball. And this, again, accommodation serves normal emmetropic eye, but it has its limits. So the closest point at which we can focus on an object is called the near point of accommodation. And as people age, this actually increases. Infants have typically very flexible lenses, and they can focus on things only a few inches away from their eyes. And then as we age, our lens gradually stiffens up, and it cannot accommodate very well. By the time we're in our mid-40s, the near point is approximately 10 to 20 inches, and people will typically have difficulty with reading. This condition is known as presbyopia. This is usually corrected with reading glasses or bifocals. These bifocals are glasses with one set of lenses for distance and another for close vision. Most people now have progressive lenses that has that smooth transition between distance and close. So where presbyopia is nearsightedness, okay, hyperopia is farsightedness, okay? This accommodation of the lens is going to depend on having the correct eye shape and proper curvature of the cornea. In a normal emmetropic eye, the length of the eyeball is normal in the anterior-posterior direction, and the lens focuses light properly on the retina. In a hyperopic individual, an individual that is farsighted, the eyeball is actually too short anterior to posterior, and so the cornea may be too flat, and the lens cannot round up enough to adequately bend the light, and so the image actually focuses behind the retina, and that causes blurry images when you're viewing something up close. And so refraction is then going to be increased by using a corrective lens, our eyeballs, our eyeglasses, a corrective lens with a convex surface providing enough additional convergence, causing that light to then focus on the retina instead of some point behind it. So yeah, this is the hyperopia perfect model. This would be where the lens normally will focus it in a hyperopic individual. Do notice that this eyeball is very short, right? You see this eyeball is very short. And so where it should be focusing on the macula lutea here in this area, it should be focusing here in this area. It's actually going beyond and focusing in the back. So all those rays that hit the macula lutea are unfocused. So then we add the corrective lens, providing additional refraction, and then that focuses the light in the proper area. And this is what? This is hyperopia, farsightedness. The opposite occurs in an individual that is myopic or has myopia. Now this is going to be nearsightedness. The distance between the cornea and the retina is too long. And so, or the cornea actually may curve too much. In this case, lenses can't flatten enough and bend the light too much, and this focuses the light in front of the retina, and it's going to blur the image when you're viewing distant objects. Myopia is corrected with concave lenses that allow for the light to diverge a little bit more before it strikes the lens of the eye. The lens of the eye is then able to provide corrective refraction. So here you see this would be a very long eyeball. Oops, long eyeball here. And so the focal point is mistakenly too far in front. And again, that scatters those light rays, blurring the image on the macula lutea. So give that person a concave lens, then it diverges those parallel light rays a bit, and then it allows the lens to focus them perfectly for in-focus vision. And what is this again? This is myopia or nearsightedness. Now I know I said what was it? I said presbyopia is nearsightedness. That was a mistake. I'm sorry. Presbyopia is not nearsightedness. Myopia is nearsightedness. And then you get into my group, the astigmatism group. Now, the astigmatism is going to occur when the curvature of the lens or the cornea is actually irregular, and so light rays aren't evenly refracted. This is going to result in blurred vision at all distances, and it's treated with corrective lenses that adjust for that person's specific abnormal corneal or lens curvatures. Okay, if the astigmatism only involves the corneal surface, they can actually undergo LASIK, or laser-assisted in situ keratomiliosis. Okay, you may have heard of LASIK. I hope you've heard of LASIK. It's a pretty common procedure now. And during this procedure, a laser is used to actually physically remodel the cornea and smooth out any irregularities in its shape. If the astigmatism is due to deeper problems, like an abnormal lens curvature, now there's actually a special form of LASIK that is able to actually induce asymmetry in the corneal surface. And so by making the cornea asymmetrical, it can actually compensate for the lens's irregularities. And I want you guys to know that LASIK is also a popular procedure for the correction of myopia and hyperopia. Just by reshaping the cornea in such a way that refraction amount is fixed and it's able to correct the focal point focusing in the retina. So we've got anatomy of the eye. You guys understand how light works. You guys now understand how light gets focused onto the photoreceptors. But what are the photoreceptors? How do they work? Well, if you'll remember, I described the retina as two layers. Okay, there's the outer pigmented epithelium, and then inside there's a layer that contains photoreceptors and other cell types that are arranged in different layers. Adjacent to the pigmented layer, you're going to find two types of photoreceptors, the rods and the cones. And these rods and cones synapse with bipolar cells, those bipolar neurons, which are going to communicate with retinal ganglion cells. Okay? And these retinal ganglion cells are going to be the most anterior cells in the retina, the ones most in the front. The axons of the retinal ganglion cells course across the retinal surface and then eventually form the optic nerve, cranial nerve number 2. And then there are also two other cell types involved in image processing. You're going to find horizontal cells forming connections between photoreceptors and bipolar cells and modulate transmit. They also work to modulate transmission, enhancing visual contrast. And you also have amacrine cells. Amacrine cells are going to be located among the dendrites of the retinal ganglion cells, and these work to respond to changes in light intensity and moving objects. Basically, they're involved in processing an image. And so here you have the different layers of the eye, of the retina, rather, I'm sorry. So going back here, you see the choroid layer, and then you have the pigmented layer of the retina. And then you have rods and cones in this area, right? Rods in this area, cones in this area, horizontal cells traveling in between them, bipolar cells. So these are the somas of those bipolar neurons. And then you have let's say a peripheral axon there and a central axon heading out. And those are going to synapse onto the retinal ganglion cells in this area. And all of those axons travel down. And so these, I'm going to say from here on, like from this area on, that would be considered the optic nerve. And this you actually can't see. This area you can't see in this microscopic image. Now, before I actually talk about rods, I do want you to remember rods and cones. You know, cones contain pigments allowing us to perceive color and high acuity vision. That's sharp vision. These function best in bright light and cannot respond at all in the dark. Rods produce low acuity vision, cannot detect color, and are unable to function in bright light. However, rods are extremely sensitive to dim light, and so our night vision is entirely almost due to rods. The difference between rods and cones explains why objects appear fuzzy and in grayscale at night. The cones are essentially non-functional, and rods produce low acuity grayscale images. But the structure of these, let's talk about the structure of the rods first. Rods have an outer segment that is essentially shaped like a cylinder. And this cylinder is actually up to 1,000 discs folding and budding from the plasma membrane, and they float in the cytosol. Okay, each disc contains a pigment known as rhodopsin inside of its membrane. And rhodopsin actually has two components. Opsin is a protein, and the pigment retinol. And retinol is actually derived from vitamin A. In the dark, the retinol is in a bent configuration known as cis-retinol, and all rods contain this pigment, and they do not distinguish between each different wavelength of light. And so here you see exactly what I'm talking about. You see all these flattened discs full of opsin and retinol in this area. So this is that exterior column that that light is going to be stimulating on. And then zooming in here, you see the flattened discs zoomed in. And then if we look inside the discs, that's when you're going to see rhodopsin made of the protein opsin and the cis-retinol derived from vitamin A within it. Now, just like rods are named because they look like rods, cones are named because, well, they're shaped like cones. This outer segment contains discs composed of the infoldings of the plasma membrane. And these discs contain the pigment iodopsin. And iodopsin is actually made of retinol and a protein photopsin. Okay? Photopsin is similar to opsin, but because of its different structure, it allows it to absorb different wavelengths of light. And there are typically three forms of photopsin. You've got blue photopsin, green photopsin, and red photopsin. And the color pigments react to slightly overlapping ranges of wavelengths. So our perception of each color is generated by the stimulation of varying combinations, of cone populations. For example, red light is going to stimulate primarily red cones, but orange light stimulates red cones as well as some green cones as well. Just like purple is going to stimulate red and blue. You guys know basic color matching. I'm not going to go into that. And so the absorptions you get here with blue cones are going to absorb lots of violets and blues and a few greens, but not very much. Rods actually don't, process any color, but they do absorb like blue light and green light, yellow, and a little bit of orange. And then you have green that mostly focuses green, red that actually focuses more on yellow up to red. Remember, red is the very far end of the spectrum that we can see. So once we get past that, we get into infrared. And infrared is invisible. But here, we have great images of, what do we have? We have cones right here, named so because it's shaped like a cone. And then the cells in between them are all rods. We have way more rods than we have cones. I think each retina has about 3 million cones, but there are about 100 million rods in each retina. So that's a 3 that's 3 to 100 mix there. And as you get away from the macula lutea, you're going to find less and less cones and more rods. In fact, there are no rods in the fovea itself. That fovea capitis, it's entirely cones. And that concentration allows that hyper focus of an object, that really HD image. And then our peripheral vision actually is due to the higher concentration of rods in the outer retina. This kind of explains why if you look at something at night, you may see an object if you look at it like just to the side out of your peripheral vision. But if you look at it directly on, that light's going to focus on the wrong place in your eye and you're not going to be able to see it. So looking just to the side of an object focuses it on the retina adjacent to the fovea, and that's where those rods are, giving you that night vision. But Chase, you say, I know someone that's colorblind. Well, that's actually a genetic defect, honestly. That person has a defective gene for one or more of these cone pigments, okay? And so that'll result in color blindness. Typically, the most common form is red-green. And this causes the red or green cone to absorb the wrong wavelength of light, and that results in red-green color blindness. These people have difficulty distinguishing red from green, and so Christmas is a real nightmare for them, with both colors really just appearing grayish brown. Now, this is typically more common in males because it's carried on the X chromosome. And so that means about 8 to 10% of males have some form of color blindness, where 1% of females have it because they have 2 X chromosomes. You have double the chance of not being colorblind if you're a female. If you're a male, you only have 1 X chromosome, so if that one has the color blindness mutation, that's all you got. Typically, this is treated, people adapt. There have also been glasses that are developed that may help people with color blindness better distinguishing between these primary colors. But most often, it's just compensation, looking for clues. Colorblind people know that the red light is the one on top of a traffic light or a traffic signal, stuff like that. The most common test for this, excuse me, is an Ishihara plate. And these Ishihara plates are just collections of dots with a number embedded using dots of a different color. So you see this plate here in this bottom right-hand corner. This is an Ishihara plate. If you cannot see the number 74 in this plate, you have a mild form of color blindness. You need to know that reliable color blindness actually requires you going to an ophthalmologist or an optometrist and actually having them test you by looking at the original Ishihara plates. This is just an image that tests you on that. If you see the number 74 in this image, your cones are functioning properly. You may, however, only see the numbers 21. 21 is not correct. There's a good sign that you may have red-green color blindness. So now let's talk about the transduction of light into an action potential. And this begins when photons from the light wave actually strike a disk in the outer segment of a rod or cone. Much more is known regarding the events in rods, so we're going to mostly focus on rods. It's probably similar in cones, but let's focus on rods. Now, the fundamental difference between transduction in the retina and in other special sensory organs, okay? Remember that olfactory neurons and gustatory olfactory neurons and gustatory cells detect smell and taste stimuli and then respond by depolarizing and signaling another cell. So that means that olfactory neurons and gustatory cells are essentially off until they're turned on by a stimulus. This is backwards in the visual system. In the dark, the absence of a stimulus, photoreceptor cells are depolarized and they're turned on. They're continuously releasing neurotransmitters into adjacent neurons. Now, what happens here is the presence of light will hyperpolarize the photoreceptor, turning it off, and that stops the release of neurotransmitters, altering the activity of other cells in the retina. And so that allows that information to be sent to the brain. The end result is essentially the same. The nervous system detects a change in the environment, and then it transduces it into an action potential. But what happens when light strikes a photoreceptor cell? We're going to need to look at a photoreceptor cell image in the dark first. So in the dark, what you're going to see is opsin and cisretinol combining to form rhodopsin in the disc membrane, right? Now, the G protein complex, transducin, and the enzyme phosphodiesterase, or PDE, are inactive in this step. So transducin and PDE are inactive. So what happens after that? Okay, sodium ions are going to enter the outer segment of the photoreceptor, depolarizing it. Now, this is all in the dark, remember? So sodium ion channels in the plasma membrane are opened by a second messenger, cyclic guanosine monophosphate, cGMP, okay? And this cGMP is bound to the plasma membrane sodium ion channels. Now, this concentration gradient favors the movement of sodium into the cell. So sodium flows into the cell, depolarizing it from resting -70 to a -40. Okay? And this shows you what I mean when I say this is in the dark. So first, opsin and cisretinol are combined, right? Opsin and cisretinol, you see here, combined. Transducin is inactive right here. PDE is inactive here. cGMP is bound to the sodium channel, and the sodium channel allows sodium to flow in, depolarizing this cell. Now, what happens in the light? Well, when light strikes a photoreceptor, the first thing that's going to happen is cisretinol changes to transretinol, and it separates from the opsin. This light is going to be absorbed by the rhodopsin embedded in the disc membrane, changing that bent cisretinol into transretinol, its straight form. Transretinol doesn't fit into opsin, and so these two split. So what happens then? Transducin and PDE are now activated. With the opsin unbound, this triggers a series of chemical reactions, beginning with the activation of transducin, which transducin then goes and activates the PDE. Well, after the PDE is active, the sodium ion channels close, and the photoreceptor begins to hyperpolarize. When the PDE is activated by transducin, PDE catalyzes the conversion of cGMP into just regular GMP. With less cGMP available, those sodium ion channels are not going to be bound and opened anymore. So they're going to close and reduce the influx of sodium ions. The membrane potential now becomes more negative than it was at rest, and is thus hyperpolarized. So here you see what I'm talking about. So light has flown in and activated. So what has it done? Well, it's straightened that cisretinol into transretinol, and opsin has kicked it out. It said, get out of here. I don't like you anymore. Transducin gets kicked off and activated, and activates the PDE. PDE actually turns cGMP into regular GMP, and that unbinds it from these sodium channels. So now the sodium channels are closed, and they kick sodium back out. Get out of here, sodium. Who needs you? Salt, get out. Okay, so that is when light hits these photoreceptors. Now, with that retinol straightened, it's now disassociated from opsin. Well, the rhodopsin is said to be bleached, okay, and it's completely unable to respond to light until it reassembles. So this has to happen through a complicated process in which transretinol leaves the photoreceptor, travels to the epithelial cells of the pigmented layer, and then there it's converted back into cisretinol in a process that uses ATP, and then gets transported back to the photoreceptor, combining with opsin, making rhodopsin again. So it turns out the pigmented layer actually forms an important part of the function of photoreceptors. Now, dark and light adaptation. Suppose you're leaving class and you see the sun is out and it's a beautiful day, and then you have to go into like a parking garage. Well, or more likely, what's happening is you're sitting in the dark and someone suddenly turns on the light. In both cases, you may feel blind at first. Your eyes gradually adjust in a process known as dark or light adaptation. These adjustments to different light intensities only partially depend on pupil size. The pupils constrict in bright light and dilate in dim light, but this is almost immediate and doesn't explain why it can take several seconds to several minutes to adjust. What actually happens here is changes in the photoreceptor cells allow for this adjustment. Adjusting to darkness depends on the rods, as they're the photoreceptors that operate in dim light, remember? In bright light, all rhodopsin in the rods is bleached, and the rods are unable to function, and vision is going to depend on the cones. When light is suddenly reduced, cones can no longer function, and so rods are slow to regenerate enough rhodopsin to function. It can take as long as 40 minutes for dark adaptation to occur and for rods to be completely functional. Now, bleaching also has an important function in light adaptation. In the dark, rods are fully functional and the retina is extremely sensitive to light. When a bright light hits the retina, then, it immediately bleaches the rods and cones, resulting in a blinding glare. And so the rods become non-functional, however, and the cones can regenerate functional pigments faster, and they're able to respond within a few minutes as their sensitivity decreases. Okay, so this is the you sitting in a dark room and someone suddenly switches on the light. Those cones are much faster to adapt than your rods are. Now, how does the depolarization and hyperpolarization of photocells affect, the bipolar cells or the retinal ganglion cells? Well, in the dark, what happens is the photoreceptor depolarizations are going to be those local potentials, right? And so action potentials in these cells are not necessary, as the cells are short enough for a local potential to spread over the entire membrane. And as that local potential depolarizes the entire membrane, the neurotransmitter glutamate is released onto bipolar cells. Now, glutamate works in bipolar cells to produce an inhibitory postsynaptic potential. You remember we talked about this, IPSPs. Some bipolar cells depolarize in response to glutamate, but they are not going to be discussed here. After the bipolar cell, after the bipolar cell is reducing its release of neurotransmitters through its IPSP, this does not stimulate the retinal ganglion cell, and so no signal is actually sent through the optic nerve to the brain. And this is in the dark, so no optic nerve signal sent in the dark to the brain. But in the light, Light is going to hyperpolarize the photoreceptor, and so it stops producing glutamate. And so that glutamate is not going to be latching onto the bipolar cells, right? Now, with the bipolar cell freed from inhibition, this depolarizes and releases neurotransmitters back onto the retinal ganglion cells. And again, remember, in these bipolar cells, a local potential is plenty enough to trigger the release of an excitatory neurotransmitter. And then when these retinal ganglion cells are stimulated by these bipolar cells, these are going to generate action potentials. And these axons of the retinal ganglion cells collectively form the innermost layer of the retina, and as they exit the eyeball, they become cranial nerve number 2, the optic nerve. And so here you have a good image, light and dark, or dark and light rather. So in the dark, this photoreceptor depolarizes, so it sends glutamate into the bipolar cell. The bipolar cell is now inhibited through that glutamate, and it does not transmit neurotransmitters to the retinal ganglion cells. Well, what happens in the light? Well, the light hyperpolarizes this cell, and so it does not transmit glutamate here. So it continues depolarizing and sending its it depolarizes then and sends those neurotransmitters across this gap to the retinal ganglion cells, activating it, and then that activation is then sent out through its axon, and once this axon leaves the optic disc, it becomes CN2. Not World War II or W2, but C, oh god, I'm just going to make it worse. CN2. So now we've detected light and we formed an image in the retina, right? That's still only part of the story. The information from the retina has to be transferred to the visual areas of the brain for conscious awareness and interpretation. And now this involves many different structures of the brain that we'll now examine. First thing we need to examine are the structures of the visual pathway. And so let's think about first the concept of a visual field. So the visual field refers to what each eye is able to see with that eye alone. However, this term also refers to what you see with both eyes open, divided into right and left with the focal point or whatever you're focused on in the middle. You should see that here. Yeah. So you see a right visual field over here, which is everything you can see with your right eye, and then a left visual field over here, which is everything you can see with your left eye. But you do see that they kind of overlap here. And then the thing that you're focusing on is actually right here in the middle. Now, the right visual field is actually shaded orange, and the left visual field is shaded purple, okay? Notice that in the center, okay, the visual field of both eyes, this is called binocular vision, okay? This area right here that is visible by both eyes, you see, is binocular vision. And then the edges are only monocular vision. And so the visual pathway is essentially this, okay? The retina of each eye detects visual stimuli from portions of the right and left visual fields. For the left visual field, these are able to be seen by the left eye's nasal retina, okay? The region of the retina nearest the nose, okay? And the right eye's temporal retina, the area of the retina nearest the temple. And then conversely, the images in the right visual field are seen by the right eye's nasal retina and the left eye's temporal retina. It makes sense if you think, where does this light draw a straight line into the eye? Okay, what happens after that? Some visual stimuli are going to cross at the optic chiasma so that all stimuli from the right visual field are processed by the left hemisphere and stimuli from the left visual field are processed by the right hemisphere. So, again, let's think of it like this. Okay, we've got nasal retina, okay, nasal retina. And then we have temporal retina, temporal retina. So the temporal retina of the left eye is going to see the right visual field. And the nasal retina of the right eye is going to see what? It's going to see the right visual field. So both of those are going to be processed in the left hemisphere. So the nasal retina of the right eye has to cross in the optic chiasma over to the left hemisphere. And then the same is opposite. That didn't make any sense. The same is true for the opposite side. The nasal retina of the left eye, okay, is able to view the left eye vision, and the temporal retina of the right eye is able to view the left eye vision. And so what happens here is the left eye nasal retina has to cross to the right hemisphere. So you see it travels down and crosses here to be processed on the right side. So left nasal processed on the right side, right nasal processed on the left side. It's really important that you know left visual field processed by right hemisphere. Right visual field processed by left hemisphere. And if you can keep that visual pathway straight in your mind, that's going to save you a lot of heartache. This is, if you don't understand it, you're going to need to draw it out. And it's going to be the only way you can understand it. Remember the structure that they cross in? This right here, this is the optic chiasma. Let's see, what else? After passing through the optic chiasma, the retinal ganglion axons are called the optic tracts. Okay, so these are now optic tracts after they've passed through the optic chiasma. Most axons in the optic tracts terminate in the lateral geniculate nucleus in the thalamus. So they go into the thalamus and terminate there, although 10% terminate in the superior colliculus or the pretectal area, both in the midbrain. And so once that visual stimulus has been taken to the thalamus, it's going to go from the thalamus to the primary visual cortex in the medial portion of the occipital lobe. And so once it goes to the occipital lobe, once it goes to the occipital lobe, the axons of this lateral geniculate neurons, they're grouped in bundles called optic radiations, and these optic radiations are going to fan out deep, okay, to the temporal and parietal lobes. And once they reach the visual cortex, each half of the visual field is now represented entirely in the opposite half of the cerebral hemisphere. So again, remember, right visual field, left cerebral hemisphere. Left visual field, right cerebral hemisphere. And so this is where we begin the basic analysis of shape, color, movement, location. And then from the primary visual cortex, information is relayed to other parts of the occipital lobe, as well as temporal and parietal lobes for interpretation and integration with other senses. If you guys don't understand this, do not hesitate to contact me. I have this crystal clear vision of it in my mind, but if you have any trouble, please don't hesitate to let me know. I'll leave these slides in. I talked about all of this actually looking at the image. I'm going to leave these in. I'll just give you a couple seconds on each one to pause and read over if you want. But again, like I said, I just talked about all this. And this is just the image. I'll leave you a clean image of the pathway. I'm not going to draw all over it. Maybe just a smiley face. But I'll leave this for you. Again, just pause this and look over it. If you have any trouble, contact me. Now, you may have noticed that a doctor will shine a light into one eye. What they're actually doing is they're testing what is known as the consensual pupillary response. And that just means that both pupils are supposed to constrict and not just the one exposed to a bright light. And this occurs because the neural connections between exist between the eyes. Instead of continuing to the thalamus, some axons of the optic tract terminate in both sides of the midbrain, this pretectal area. And in this area, these pretectal neurons contact other neurons that send their axons back through the oculomotor nerve to the pupillary muscles. And since signals about bright light hitting the retina in one eye travel to both sides of the brain, this reflexive constricts the pupil in both eyes. Clinically, this is very important. If this does not occur, there's either damage to the retina, the optic nerve, or the individual's brainstem. And as I discussed in the brain chapter, you need your brainstem. It's very important. Because our eyes face anteriorly, so much of the visual field for one eye overlaps with that of the other eye, and that gives us that binocular vision, right? Well, each eye sees many of the same objects, but from a different viewpoint, and the brain is able to compare these images and use the differences to create stereoscopic vision, producing our depth perception. The distance of an object from an eye. And the loss of vision in one eye will typically destroy the ability to compare images, and so depth perception or stereoscopic vision is lost. Now, the act of seeing is obviously more than just detect image, be aware of stimulus. The areas of the brain are more involved and more complicated and very much more extensive than what has just been mentioned. The primary visual cortex receives axons from the lateral geniculate nucleus, and the retina is now mapped on these areas, actually. However, the image can actually be distorted. Parts of it falling off the fovea of the retina take up much more area than parts from peripheral areas. Okay, so this occurs because of the increased numbers of photoreceptors in the fovea, and those increased numbers provide enhanced sensitivity. Much like the sensation in the tip of your fingers, it occupies a disproportionately large area of the cortex compared to the sensations from your calf. I want you to think about that. So, in addition to conscious awareness of visual stimulus, the primary visual cortex is also going to be that initial processing location for movement, shape, color, and general location of an object. And then it combines the images from each eye for binocular vision. Then it's sent one of two general pathways. There's the dorsal pathway and there's the ventral pathway, also known as anterior and posterior pathways. I'm sorry, posterior is dorsal and anterior is ventral. That's the same terms, just they mean the same thing, they're just different terms. The posterior pathway, or the dorsal pathway, involves the parietal lobe, and this is essentially going to be interpreting motion. A person with damage in this region may have difficulty pouring coffee into a cup or crossing the street because they're not able to detect the movement of the coffee or the cars. Okay, the ventral pathway is actually specialized for processing color and form of objects. some of these neurons respond more vigorously to faces as opposed to furniture or skyscrapers. And so those people that I may mention before suffering from prosopagnosia, this is facial blindness. These are people that cannot recognize even close family members by their face. They have to rely on other cues like the sounds of their voice to ID these people. Now, damage to this pathway, this anterior pathway, can also result in loss of color perception, despite the fact that functional cones are present. And this rare condition is called achromatopsia. And this, this could be more unpleasant than you expect. How appetizing would you find it if someone poured a bunch of black ketchup on your gray French fries? It wouldn't be very great. So let's put it all together. Imagine you're sitting on the beach, you're watching a friend surf the waves, and then, oh my God, they tumble off their surfboard. How are you seeing this step by step? Okay, the first thing that happens is light that gets reflected off of your friend and his surfboard gets transmitted back into your eyes, right? Well, the cornea and the flattened lens provide enough refraction that focuses the light rays from from your friend and his surfboard back onto the retina, and that's because the subject is more than 20 feet away, right? Then what happens? Well, those light rays are going to hyperpolarize the photoreceptors, allowing those retinal ganglion cells to send action potentials through the optic nerves and begin actually sending signals, right? So those action potentials get sent out through the optic nerves. Well, which nerves? Okay, let's see. This is the image from your right field gets processed in this, gets transmitted from this area and this area, right? Anything over in this area gets transmitted just like that, right? And so they both have to get processed on the same side of the cerebral hemisphere. So in the right eye, they cross over to the left hemisphere, to the thalamus, and then eventually back to that primary visual cortex. Let me change color here. The images, oh God, that yellow is not going to show up. The images from here, all right, these are going to get transmitted over here and get transmitted by those retinal cells. Well, they are going to go down and, these don't cross, right? But these do. The ones in the left side cross for the left visual field. And so now those are all being processed on the right hemisphere, okay? And so this is actually what you end up with, this right visual field image and your left visual field image, but your brain then flip-flops them. So that is how you interpret. That is how vision is interpreted, all the way from light to action potentials to cerebral integration. This is actually the end of part 1, okay? This was quite a bit of it. We'll come back and we'll talk about especially hearing, and then balance. Oh gosh, and there's one more, isn't there? I know there's one more. Okay, so after this we've got anatomy of the ear, physiology of hearing, vestibular sensation, and then after all of that, how these special senses work together. So that's it for this one. I will see you in the next one.